I'm John Lyons from the University of Wisconsin Zoological Museum in the state of Wisconsin in the United States. I want to echo all the previous speakers and say what an honor and a pleasure it is here to be here. It's ever since the Goodeid Working Group started 3 years ago, I've been very intrigued and wanted to meet a lot of the people and see what was going on firsthand, so it's really great to be able to be here and put names to faces, as we say, and get a little better handle of who's keeping what and who's doing what. So it's very exciting. I'm very glad to be here. Unfortunately, my talk, I think, is going to be kind of depressing because I'm going to be mainly documenting some very serious and very disturbing declines and, in some cases, disappearances of many of the fish species that we like so much. But hopefully it will also be somewhat maybe inspirational or an incentive to continue to do and maybe increase doing what you've already been doing so well in terms of maintaining many of these species and keeping them alive and working together and trying to develop networks so that we can assure that these species can survive, at least in captivity, and hopefully we can do some on-the-ground conservation and bring them back in the wild as well. But what I want to do is I want to talk about what is the status of all of the Goodeids, all the species, in the wild in Mexico. And so what is their condition right now? What are their prospects for the future? And talk a little bit about why they've gotten to where they are today. And then hopefully end up with, on a little bit more positive note, talking about some of the things we might do and some of the restoration or rehabilitation things and practices, conservation things, to help preserve them and hopefully recover them a little bit. So for this crowd, it's easy to talk about how exciting and cool and fun Goodeids are. You don't have to explain, well, they're pretty and they're biologically interesting. Everyone in this room, I think, is already very interested in Goodeids. And I first began working with Goodeids about 25 years ago in Mexico. And not only did I fall in love with the fish themselves, but I also fell in love with the places that they lived. Because really, many of the Goodeid habitats, as you've seen earlier in some of Juan Miguel's presentation, some of Yishu's presentations about the habitats they live in, they're beautiful. And they're really interesting, wonderful places. And so, you know, not only is it fun to see the fish, it's fun to go and visit these places themselves. But unfortunately, as all our previous speakers have alluded to, many of these habitats are in trouble. The Goodeids share their habitat with the most populated and rapidly developing area of Mexico. And so there's many issues in terms of human uses and human demands on the resources that make Goodeid survival difficult. We've heard about, for instance, Juan Miguel talked about a lot of systems drying up, and I like this example. This is Lago de Chapala, which is the largest natural lake in Mexico. It's on the order of 40 kilometers long, 15 or so kilometers wide. And this is how it should look, but because so much water has been extracted and diverted for irrigation, for industrial uses, for municipal uses, this is how it will look anytime there's a drought. So, I mean, you literally lose kilometers of lake surface area. You can barely see the water there out maybe 5 kilometers from the dock at Chapala. And so, obviously, when you have these sorts of losses, it's going to affect the fish in bad ways. You also have terrible pollution problems, as again, we've seen examples of, and these are just, sometimes it's really not very appealing to go into the water to try to catch fish because it's so polluted. And then finally, as everybody has talked about, but I think Sean illustrated particularly well, is the whole problem with invasive species, exotic species. And this is just a handful of the many, many species that have been introduced either intentionally or accidentally in central Mexico, and many of these have had all sorts of negative consequences, and we'll talk specifically about some of those in a moment. So you've got all these things happening, and it became clear then, you know, with all of these threats, that a number of us in the United States and Mexico who've been looking at Goodeids we're seeing all sorts of species decline. We go to habitats where a few years before we'd seen lots of individuals and we wouldn't find them. And so back about 2000, Omar Dominguez, another Mexican colleague, Norman Mercado, and myself tried to sit down with help from other colleagues in Mexico and really lay out exactly what the status of the Goodeids were, what their abundance was, what populations still existed, what populations had been lost, what the trends were. And it was published in the first proceedings of the first Viviparous Fishes Symposium in 2005 as this article, Conservation Status of Mexican Goodeids, Problems, Perspectives, and Solutions. Now this was published in 2005, but it refers to how things were about the turn of the millennium, about 2000 or so about 10 years ago. And unfortunately at that point things were not good at all. So we were seeing more than half of the species, half of the species were endangered. And by endangered, that means they are on the brink of extinction. They have almost disappeared. 2 species actually had disappeared at extinction. 13 were vulnerable, which means they're on the brink of becoming endangered. And only 3 species, less than 10% of the fauna, were really what you'd call low risk, or that had some long-term stability or potential to persist. So over 90% of the fauna was either lost or in trouble. And so this was very, we knew things were bad before we started this, but when we actually did the analyses and did the calculations, this was very, very disturbing and really concerned us quite a bit. Well, this survey has engendered a lot of attention and hopefully focused a lot of different aquarists and conservation groups on particular species and raising them and so forth. But unfortunately, over the last 10 years, it's been clear that things are not getting better. In fact, they're probably getting worse. And so what I want to talk to you today is what the status of Goodeids are in 2011, essentially now. And so updating that 2001 survey that we did and talking specifically about what's going on and what some of the issues are. And some people said, well, you just did this survey. Why do you need one so quickly? Well, anytime you're dealing with rare fish, I think it's a good idea to do regular quantitative scientific inventories and really understand where they are. So that's just a good idea. There have been some minor changes in some of the conservation terminology in terms of what we call things. And so there's a need just to sort of bring things up to the most recent language. There's also been some important taxonomic changes in the Goodeids that have some implications that I'll talk a little bit about. But mostly, as I said, they've continued to decline. Things have continued to get worse. And so, what we looked at 10 years ago is actually better than things are today. So this is what we have again today. So there's still half the fish in danger, but now we've gone from 2 extinct to 5 extinct. And I'll go through all 5 of those in a second. And we have actually fewer vulnerable fish, but it's because some of the vulnerable fish are becoming endangered, some of the endangered fish are becoming extinct, and so everything is not even slowly, rapidly getting worse. And even within some of these categories, there are fish that 10 years ago just barely qualified as endangered, and now are critically endangered, are one population away from extinction. So things are really not good. Just to show you the actual numbers, this is what we had in 2001. These are the numbers. And again, there's some taxonomic differences so that these don't exactly match up. This is what we have in 2011. And just for contrast, this is the official Mexican government list of threatened and endangered species. And you can see that there's a major difference. They have many fewer endangered and threatened species and many more species that are considered low risk. And that is not necessarily because the Mexican list is incompetent or bad or something. It's just that because it's a political process, it's a legal process, it's very slow. And so even though the list itself in Mexico was published in 2001, it really reflects what conditions were like in, say, 1985 or 1990. so more like 20 or 25 years ago. And this is what things were like then. So this gives you an idea of how quickly things have changed. So 25 to 30 years ago, most Goodeids weren't in trouble. There were certainly some, but many fewer than there are today. And so the process of decline has been, unfortunately, accelerated. It's becoming worse and worse. And even, like I said, even within some of these categories, many fish are still decreasing. So they maybe haven't changed their category in the last 10 years, but they're continuing to decline. So again, more than half of the fish, 2/3 of the species are declining within the last decade. So things are really not good. So what I want to do now is go into those. We have those specific numbers, but they're numbers, and which species are in each of these categories, and which species are declining, which species are stable. I want to talk a little bit about that. I should also say that I did put together a list that has everything that was on the back table. I don't know if it got buried in the... But there, I did sort of put the list together, exactly which species and which category and so forth. So I'll be flashing through some of these, and it'll be hard to keep up. So but there is a list back there, so you don't have to try to write all this down. But let me just talk about how I came up with these numbers, and generally how we get to what I've been talking about. First of all of our conservation categories are based on the International Union for the Conservation of Nature, the IUCN, which is the international standard for rare organisms, both plants and animals. And so they have very specific criteria and nomenclature for how things are designated and what qualifies or what would cause you to put a certain species in a certain category. Some of those criteria have changed a little bit since 2011, or since 2001, so I've updated those. And so this is the standard upon which international conservation is usually based, and so that's what we've tried to use. I should point out, however, these are not official IUCN designations. We have not taken the next step of actually applying to the IUCN for official designation of each of these species. A couple of reasons. One is just it's a lot of work and it's a lot of time, and it's just a question of how many hours are in the day. But the other is, and this is more of a philosophical issue, is when things become listed officially by something like the IUCN, they get on the IUCN Red List, for example, it becomes much more difficult to move them about, to have hobbyists legally carry them from one country to another, or exchange them among themselves, or even keep them in their own fish rooms legally. And so there's this sort of idea, is it better to have them designated officially so that will become more maybe globally prominent in people's eyes, or to kind of keep them unofficially designated so we still have the flexibility then to move fish around and keep them without all sorts of complicated paperwork and bureaucracy. And for the moment, it's been sort of the impression of most of the people working on Goodeids that it's better to keep them unofficial. And I think Sean can tell you, having anything officially listed in the United States makes doing any kind of conservation work for them very, very complicated. And the same would be true if they were IUCN listed officially. And I should also point out they're also not officially listed by the Mexican government. So these are, my opinion, basically when it comes down to it, they don't have any sort of official legal standing or anything like that. But let's start from the worst and work our way to the best. So extinct. That is, by IUCN terms, when there is no reasonable doubt that the last individual has died. There are no individuals in the wild, there are no individuals in captivity. 2 species fit that right now for the Mexican Goodeids. One, I think most people are aware of, Carcharodon garmani. This was a Carcharodon that was described over 100 years ago and has never been seen since. It occupied a small valley that was heavily, heavily modified almost immediately at the time when it was described, and people have looked for it for 100 years and not found it. And so that's long been known to be extinct. But the other one I think that will surprise people is Jarndyctes, formerly known as Hubzini turneri. And this is extinct, ironically, because of a taxonomic change. This species traditionally was thought to have occurred within two different places, the Lago de Cuitzeo, Rio Grande de Morelia area near Morelia, and that area has been heavily, heavily modified, and there have been no Huxina observed there, no Giardinichthys turneri observed there since the 1980s, despite lots and lots of looking from lots of people. So no one doubts that they are actually no longer present in this area. It used to be thought that they occurred also in Laguna Zacapa, which was part of the Lerma system. But in 2003, this population was actually described as a separate species, Giardinichthys iraniae. And so, and there's some dispute, some ichthyologists don't accept this as a separate species, but if you accept this as a separate species, then this population still persists, but it's now a new species, meaning that Turneri is strictly what was here, and that has been lost. And so that species is now extinct. There are no, to my knowledge, and I would love someone here in this audience to correct me, but to my knowledge, nobody has any captive populations of Giardinichthys turneri from Cuitzeo or the Cuitzeo system. And all the so-called turneri that are there are actually from Zacapu, or the vicinity of Zacapu, in which case they are now actually called Iraniae, and they are not then the same species. So that species then would be extinct by that definition. Extinct in the wild, when it is known only to survive in captivity. We have three species here, and I want to go through all three of these because all three of these have changed in the last 10 years or so. The first one is Scyphia francesa. That was, as traditionally recognized, last observed in the 1970s in the Tiuchitlan springs in the Upper Emeka Basin. But there's been a lot of talk that there's actually a new population out there that represents Scyphia francesa. And so I'll talk about that in a second. Alodontictes polylepis. That was last observed in the wild in 2000, and so for our 2001 list, we considered it still extant, but it appears to have disappeared since then, primarily due to habitat loss, and I'll talk about that. And then Alatoca goslini also was present as of our 2001 list, so we had it listed as critically endangered, but now it appears two have disappeared, primarily due to interactions with non-native fish, and so I'll talk about that. So let's talk about Scyphia francesa. The way it is described in the literature, the way it's always been thought of, Scyphia francesa was a species endemic to the Tiuchitlan springs in the Upper Emeka Basin. By the time it was actually described in the 1970s, it had already disappeared from that system, but there were lots of captive populations, and they're still widely circulated, still in existence. But then, in 2008, Omar Dominguez found a new Scyphia in a tiny, and by tiny I mean something the size of this room or smaller, pool called the Cayucapan, I call it Cayucapan Pool or Cayucapan Springs, in the adjacent but separate Sayula Lake Basin. And what was interesting about this species is when you did the genetic analysis, it was essentially identical to Scyphia francesa. So the initial thought was, hey, there's a new population out there. And, maybe Scyphia francesa really isn't extinct in the wild. Well, this is the Cayucapan Scyphia, wild-caught fish, freshly preserved wild-caught fish. And this is what they look like coming out of the wild. Like I said, genetically, they're essentially indistinguishable from the Scyphia that were found, at least that descended from populations in the Tiuchitlan springs. And so by that perspective, then maybe these are Scyphia francesa. However, in terms of pigmentation and some of, sort of first glance morphological characteristics, they actually look a little bit more similar, to my mind and to a number of other people's mind, to Scyphia multipunctata, which is, again, a closely related species in the lower Lerma system. And so, certainly there's been no exact resolution of this question at this point. There's no consensus among people who are thinking about this. But since this is my talk, I get to give my opinion. And my opinion is that I really think that it's neither, and it probably would warrant being being described as its own species. It certainly is genetically like Francesae, but morphologically it's pretty different, or pigmentation-wise it's fairly different looking. But it doesn't look exactly like Multipunctata either. If you accept my view, then Scipio Francesae is still extinct in the wild, because this isn't Scipio Francesae. Unfortunately, the Cayucupon Springs completely dried up in 2010, and as far as we know, everything there died and disappeared. Fortunately, Omar took a captive population to Morelia. In that population, I can see there's specimens downstairs. There's specimens in several places. But then by that, then we have another extinct in the wild Gudea, if it is indeed a separate species and it is indeed eliminated from Cayucupon. So it doesn't really matter what you call it at this point. It remains extinct in the wild, and all that is left are captive populations as far as we know. Alinotectes polylepis, a species limited to the upper Ameca River, a handful of streams. This is a species that likes current, likes fairly fast water. This species appears to have disappeared, again, because of long-term human impacts that reduced water tables, reduced water levels, then couple that with a natural drought that completely dried things up and basically pushed the species over the edge for the final time. This is a species, like I said, that likes fast current, like shallow riffle areas. These are the first areas to disappear as the stream dries up. There was a major 2001 drought, the beginning of a series of droughts actually, in that area where it occurred. And these are the last two places, the De La Pola River and the Diablo stream, where it was captured back in 2000. And there's been none found in at least 4 very targeted, thorough surveys since then, with electroshocking gear, with nets, with everything we can use, and have been unable to find these in extensive sampling. So, again, we fear that this species has disappeared, primarily due to habitat loss. Its habitat dried up. And then Alitoca goslinii. This also in the upper Ameca River, it actually overlapped slightly with the range of Alinotectes polylepis. This species appears to have been driven extinct by a non-native competitor predator. It was known only from this one tiny little stream, the Potrero Grande in Jalisco, which is so tiny when you drive over on the road you don't even know it's there. It's just bushes and things. You can't even see the water. In 2000, the population there was doing fairly well. It was a single isolated population, but it seemed to be doing well. By 2004, Xiphophorus helleri, the common green pet store swordtail, had invaded the stream, and these are the numbers from our standardized surveys. So in 2000, there were lots of Alitoca and Xiphophorus, and you can see how things changed. And there have been subsequent surveys since 2007, and no one can find the species anymore, and all they find are hundreds of Xiphophorus. So again, we fear this one is gone. And this one, I don't know if there's maybe one other than Morelia. I don't know if there's any other populations. Does anybody here have goslinii in their? You do?
Yeah.
Oh, good. Did you get it from Morelia? People in Morelia?
From Germany originally.
Oh, the original one from, okay. So the older one, yeah. So that's good. So there's at least one in Europe.
There are many of them, yeah.
Oh, good.
Different countries. I think we have them in 7 or 8 countries.
Oh, very good. Very good. I didn't realize that, because it's very scarce in the U.S. I don't know maybe 1 or 2 people who have it. At least they could talk to me. So maybe more do, but that's all I know of. So good. Well, that's good news. Okay, now we'll just go to critically endangered. And again, there's a series of quantitative criteria here that are used, and different species meet different of these criteria depending on where they are. But this has the dubious distinction of being the biggest category of Gudea. It's 16 species. They're all listed here. And I won't go through all of these, but I want to pick out a few that are relevant and talk a little bit more about them in detail. One area of what you call good news is that Ameca splendens doesn't appear to be quite as rare in the wild as we once thought. Historically, this was always thought to be limited to the Teuchitlan Springs that I've already mentioned. But again, thanks to the work of Omar Dominguez, in 2007, 2008, two more populations were found. One in the El Moloya Springs to the west of Teuchitlan, and then in the Cayucapan Springs that I've already mentioned to the south. So that little springs that has unfortunately dried up had two, distinct, very important populations, as well as a couple other Gudeids on top of that. So a very interesting place that sadly lost. One interesting thing is the Teuchitlan Springs I'll show here. Some of it is natural, but some of it is very modified, and it's actually what's called a balneario, a swimming area that people pay money to go and swim on hot days and picnic and so forth. And the best populations are actually in this actual swimming pool. So you can pay money and swim with the Ameca, sort of like swim with the dolphins, swim with the Ameca. This is the El Moloya Springs. It's also a semi-natural balneario, very small. And again, the Cayucapan Springs, also very small and which has disappeared. So there's one more population of Ameca out there, so that's good. Certainly these two populations should be treated as distinct entities and not mixed in terms of captive rearing. Zogoneticus tequila. There are persistent rumors that this species is completely extinct in the wild, and I'm happy to say that's not true. It still persists, but the bad news is the population is so tiny that it could disappear tomorrow. It too is endemic to the Teuchitlan Springs, and no one could find them there for many years, but then, I can't remember if it was Omar or somebody, stumbled upon a little side springs in this complex and found them. But this is a tiny spring. This is how big it is. You can see it's maybe a quarter the size of this room. And maybe there's 100 adult fish there. And so it's extremely vulnerable. And so there's no guarantee that this species will live from day to day. So we can almost treat it as extinct in the wild. And this population, there's been some analyses done, it's extremely inbred as well. So that doesn't bode well for long-term survival. Neophorus regalis, or sometimes called Alatoca regalis, this is another species that's declined precipitously in the last 10 years and gone from being merely just on the edge of critically endangered to being on the edge of actually extinct. It's found in the Los Reyes Valley in the upper Balsas Basin. In 1980, it was known from 5 distinct areas, all of which had fairly stable populations. By 2000, it had shrunk to 3 areas, and by 2010, it was in 1 area, and that population was very small. So the Los Reyes stream, which used to be the stronghold for the species, you can see, and it was present there in good numbers in 2002, you can see by 2008 it had been modified into a concrete ditch for irrigation, and so there were no longer any Alatoca left there. Cuitupan River in Jalisco, that still has good habitat, and there were fish there, Alatoca there in 2004. By 2010, they disappeared, presumably because Zophophorus helleri again had appeared in the system and taken over. And the only place the species still persists is a little tiny stream called the Guatarillo stream in the little town of the same name. It's present there as of 2010, just a handful of fish. And this is right in the middle of the town. It gets all kinds of runoff and debris and pollution from the town. So it's hardly a good long-term prospect for this species. And then finally, Yishi already talked a little about Alatoca mikai, and I know Juan Miguel was asking about this. So we've had the same data that Yishi talked about. This species is really almost gone. It's endemic to the Lago Zeroin Basin. It used to be found in the lake itself and in its tributaries. The largemouth bass, Movina negra, Micropterus salmoides, entered the lake during the 80s, and this is an efficient piscivorous predator. It ate up all the Gudeids in the system very quickly. They were gone from there by the early 90s. And it was limited then to Lake Popopayo, which is a little, as the issue showed us, was a little isolated former tributary system, many kilometers away. But by the mid-2000s, largemouth bass, micropterus had entered Lago Popopayo, and really the only place they live now is this little outlet of the system, which is a little tiny stream. Again, runs right through the middle of a town. Women wash their clothes in this stream, and all sorts of soaps and so forth go into it. And we're just talking about a handful of fish that we could find. So the long-term prospects for this species, again, seem very bleak because, again, an exotic species has taken over its habitat. Endangered species. We're getting better here. These are a little bit bigger populations, but again, they're still endangered. 7 species on the list here. Again, I'll just have one example here, and this is another one where some taxonomic changes, our better understanding of the evolutionary relationships, have actually pushed a couple of species into the endangered category. And Zogoneticus quitsioensis in 2000 was considered a single fairly widespread species. It was classified then as vulnerable. It was found fairly widely in Chapala area and the Lerma system and adjacent areas of those two systems. But then in 2008, it was split into 2 species. The western part were called Zogoneticus parapachus, and the eastern part retained the name Zogoneticus quitsioensis. And by that split, you basically cut the populations in half, and just by that split, they became endangered. They met the endangered criteria. But it's not just a sort of a bookkeeping bureaucratic thing here. They also were declining, and so during the same period, both species lost populations and lost abundances. So it's not only just a taxonomic shift, it's an actual decline. Vulnerable, these are fish that are on the brink of being endangered. Again, a long list here. 9 species. I'll talk a little bit about Chapalichthys incaustus in Lago Chapala, which is the center of its range. Here's a question where, again, non-native species have taken this species and it caused it to decline. It hasn't changed its category. It's vulnerable now. It was vulnerable 10 years ago, but it's much less common than it was 10 years ago, and it's closer to moving into that endangered category. So in 1990s, despite all those water level problems I showed you in the very beginning, there were lots of Chapalichthys incaustus in Lago Chapala. You'd go out and pull a seine, and you would always catch several, and often hundreds. So they were one of the most common and widespread fish in the lake. But then by 2002, non-native piscilids came into the system, probably mixed up with introductions of tilapia, which is one of the dominant fisheries species in this, another exotic. And they came in and they reached incredible numbers, so much that the local fishermen refer to them as a plague. They're so common. And as far as we can tell, then they have really hit the Chapalichthys population, so that now Chapalichthys is still there. It hasn't disappeared from the lake, but it's relatively uncommon. It's infrequent now to see that species when you're out looking and seining and looking around. You don't see it much in the fishermen's catches. So they've really gotten uncommon just in the last few years, presumably because of these piscilids. And then finally, the least concerned category. These are the only 3 species that seem to be in reasonable shape. All of them are still fairly widespread, still fairly common, but all of them are still also declining. They haven't reached that vulnerable category, but they're all less common than they were 10 years ago. So, all that bad news, let's talk about what maybe we can do, how we can make things a little bit better. The good news is there's still a few places in Mexico where you can go and see scenes like this, where you have 4 or 5 Goodeids in large numbers just swimming around in front of you, and it's really, for a Goodei lover, it's paradise. You just look down and see all these different individuals and different species, and they're breeding males, and you can watch them. And it's great. And this is La Luz Springs in Michoacan, which is a very nice place to still see Goodei. It still has healthy populations of these species. Well, three things that are critical if we're going to conserve wild goodeias. The first is to protect the best remaining habitats we have left. There are, like I said, there are still some good places. These species still hang on in places. So we really need to make a strong effort to protect what we've got left. And the species that tend to live in spring-like habitats, what I call the spring species of goodeias, are the ones that are probably going to be the easiest because they live in very compact areas where conservation and land control and determining what people do or don't do in the area is just logistically much easier. Probably the nicest and most important system still left is the La Angostura Springs, which are a little sidearm of Laguna Zacapu. They have 7 different species of goodeias. So if you can ever visit the Morelia area and get to Zacapu and see this area, it's just wonderful. It's full of fish, and including some very rare types of species. La Metzita Springs near Michoacan is almost as good with 5, another wonderful place, but heavily encroached by human development. So again, not by any means secure at this point. And so the key should be to secure these places, to preserve them for the future. And there's others shown here. There's a place called Lagos Negritos near Chapala that still has sort of what Chapala used to look like. There are some of the springs in the Durango Valley where Paracadon occurs. This is 27 November spring system, which still has fairly good populations. This not very appealing stock tank in the middle of nowhere at Hacienda San Sebastian has some interesting fish, including this new Xenotoga eisneri that Kyle will be talking about next. And so that's an important area. So there's a lot of these areas to be protected. The good news is when you're trying to protect springs, there's lots of reasons why you would protect them besides just goodeias. And so these are reasons that are going to resonate with people who don't really care about fish. So they can be used for drinking water, livestock, or irrigation water. They have value for recreation, for swimming, and so forth. And so that makes them an easier conservation target. And indeed, many of these springs already are preserved to a more or lesser degree for those very reasons. So Copachuro Springs in Michoacan, that's a municipal water supply that has 4 different species of goodea, a really beautiful spot. And it's fenced off. They don't let people dump stuff in there. There's regulations about land use in the immediate vicinity and so forth to protect their water supply. But what people often don't really think about in terms of protecting these systems is the whole issue of non-native species. And so on the one hand, where no one would think, oh, I'm going to dump a can of oil or paint into this water because I have to drink it, they have no compunction about, well, I'll throw some tilapia in here, or I'll throw some carp, or I'll throw some catfish. And so this particular system already has 3 different exotic species and is vulnerable at any time to more coming in. And they alone, the habitat could remain excellent and the goodeias could be eliminated if the wrong exotic got into the system. If you're dealing with riverine goodeias that live in river-flowing water systems, then conservation becomes trickier. They have much larger areas that need to be protected because they need the entire watershed under some sort of control, not just the immediate area around the spring. And here's a couple of examples. These hubsides and Athena Resilene, these like fairly large, fast-flowing river systems. This is the approximate range of most of the riverine goodeias. It's a fairly big area. And at this point, there's only one reserve that really does anything towards protecting those. That's the Sierra de Manatlan Biosphere Reserve that protects 3 of those species through land use controls and through controls on pollution and so forth like that. But the other 4 species that are considered riverine goodeias have no protection whatsoever. And so a big challenge will be trying to figure out how to protect big areas of land in an appropriate way to protect these sorts of species. That won't be particularly easy. Now, in addition to conserving the good habitats left, there is also the potential to actually restore habitats. And Sean talked a lot about restoration for the Phrynixes and Empetrichthys. But it's also potential for some of the Mexican goodeias. And I want to show somewhat of a success story that we've had in restoration in the Ayuquila River, which is in the Sierra de Manatlan, on the edge of the Sierra de Manatlan Biosphere Reserve that it's mentioned. Now in this area, it's a big sugarcane area. That's the major crop. The sugar mill that processes the sugar cane is the primary employer, the primary source of economic activity. And prior to 1999, it discharged all of its untreated waste directly into the Ayuquila River, which was a major fishery for local people, a major source of biodiversity. And there was just gross pollution, no dissolved oxygen in the water, and no fish for long stretches. And so here's some actual data on that. This is river kilometers. This is upstream to downstream as you go down the Ayuquila River. And then this is dissolved oxygen in white, the amount. And you want to have, ideally, at least 5 milligrams per liter to support healthy fish populations. And then here's the number of fish species present. And so you see lots of oxygen upstream of the sugar mill discharge, which is right here. And as soon as that comes in, there's no oxygen for about 20 or so kilometers and no fish for 25 kilometers. So completely devastated, and it's only until you're about 40 kilometers downstream that you see recovery. So a big chunk of this river lost into the sugar mill discharge. This was very contentious in the area, a lot of protests, a lot of back and forth between the fishermen and the cañeros who cut the cane, and there was some violence even over this. But finally, in 1999, they came up with a sort of a compromise plan where the sugar mill's waste were now, rather than sent directly to the river, they were sent into irrigation canals for 8 kilometers before they entered the river. And during that process, there was some natural decomposition of the pollution. There was some re-aeration of the water. And so by the time the canals hit the river itself, the river wasn't as polluted. And so you can see this here. Here is the old sugar, where the old sugar mill discharge used to be. And now you can see the dissolved oxygen is good immediately below that. Here's where it comes in now after going through the irrigation canals. We see a big oxygen sag, but it doesn't go all the way to zero. There's still a little oxygen in the water, and it recovers more quickly. And the fish population is impacted, but at least some fish survive in the area. And then, again, their recovery is more quickly. So now in an area where we saw no fish whatsoever, we see lots of Iliodon furcitans, which is a particularly tolerant Gudiid in that area. So, good news, right? Well, one of the things is every time you solve one problem, another problem comes up. And in the process of cleaning up the water here, you made the oxygen better, made the conditions better for the Iliodon. And, it's just a nicer stream now. But you also made the water better for Micropterus salmoides, the predator. And so they became more common, and they began to eat the Iliodon. So in some areas, you have at least a few Iliodon now, but you have many fewer than you would expect just because there's good habitat for this exotic predator, which is suppressing then the Iliodon recovery. So certainly restoration is a complex and difficult process, but it needs to be tried. Finally, there's the whole issue of near and dear to everyone's heart here of establishing and maintaining captive breeding colonies. The Fish Ark facility in Michoacan is certainly critical to that effort. The work being done here at Chester Zoo, some other places, and certainly by all of you in this room is essential to this. And really, this is a cornerstone to any sort of conservation project. So I applaud all the efforts of everyone in this room. It's critical and critically important. This is my list of my sort of, if you had to prioritize the fish that are there, these are the ones I would say would be most important, the ones that are most imminent, either disappeared already or most imminent disappearing. But bottom line is any Gudiid you can keep is good. I mean, they're all in some sort of trouble. Even the Iliodons and the Gudias that are still fairly common, they're declining. And 10 years from now, I might be pushing them into some of these endangered lists at the rate they're going. So all of these fish weren't captive, rarely. So, sum up. Wild Gudiids in serious trouble. Most are endangered, most are in decline. The primary threats are the water quantity issue, habitat, water quality, and non-native species. Conservation is a 3-step process. Protect the good habitats, do restoration where it's practical, and continue and maintain captive populations. And then the Gudiid Working Group and other sorts of conservation groups and you as individuals certainly can play a really key role in this by, first of all, educating the public and advocating for Goodeid conservation, which the House of Emirs is doing as we speak, continuing the captive rearing, which you're all doing, and working on raising funds, raising support to actually do some of these on-the-ground conservation projects. Protecting land, restoring areas, it costs money, and there's no way around it. There's lots of advocates for pandas and for tigers and things like that, and there's money for that sort of stuff. There isn't much money for fish conservation, and so this is where this group can play a role. And even small amounts of money, judiciously used, can have a big impact on some of these key habitats in Mexico. And so with that, I'm happy to answer any questions.
I have one question.
Yes.
The old Scyphia francese, does that still exist in captivity?
Yes, it's actually fairly common. There are domestic populations, a lot of people. So that's been in captivity now for 30 or so years, and those populations have persisted. The new Scyphia, the Cayacapan Scyphia, I guess you have some, and Omar has some, and I don't know, you have some.
In the Netherlands we have them too, but we don't have the old one.
Well, the old one is still available.
How do the swordtails get spread around in like these smaller habitats?
I think it's aquarium releases. You see those swordtails for sale in pet stores in Mexico, and guppies. You see guppies all over the place. You see armored catfish. You see a long list of common aquarium species. There are some areas in Mexico as well where they're actually raised for sale. So there are tropical fish farms, and the Balsas, upper Balsas Basin, that's been a huge problem. There's an industry in the upper part of the basin, and all of the convict cichlids are all over the place. So all of the common ones, they've escaped or been deliberately. And then once they're in a system, they are amazingly good at spreading around. So Portero Grande, I can't imagine someone dumping something in this little creek, but it flows into the Ameca River, which has lots of swordtails, and I think they just work their way upstream.
Is there any strategy that you favor to protect the spring habitats?
Well, it's going to be different in every case, but it definitely requires local participation and a convincing or working with local people that it's in their interest to protect this spring. And like I said, many of these are very important for drinking water, for irrigation water. They're popular recreational areas. So in that sense, it's not difficult to work with people and have them say, yes, we don't want people to pollute this. We don't want people to degrade this habitat. The issue is the invasive species. Either them invading on their own, getting in there on their own, or misguided people just dropping them there, because most people don't see that as pollution, even though it is. It's biological pollution. But the average person thinks, oh, I don't want to kill my pet. I'm going to do this humane thing and let it go in the springs here, and that will be good. Or, I want to go fishing in the spring, so I'm going to put these lobina, these bass, or these trout, or whatever it is, in the system, so I can catch them later, and this will be good. So when I come here to have a picnic, I can go fishing. And so this is really a struggle, and that's going to be the hardest part of that. But it's education, working with people, and so forth. Thanks.
Thank you. Shall we go on immediately with Carl, or just a few minutes break?
The invitation and the support to come here. It's great to have the opportunity to come speak to a group like this on some research that I've been doing the past couple of years. I will say that I'm relatively new to the Goodeid world. I believe I probably collected my first Goodeid in about 2005 with John when we went to Lake Chapala. Actually, my initial interest was to go there and collect silver signs for a grant that I had gotten funded. And sort of as a bycatch, we started getting Goodeids and talking with John, and became very interested in addressing some population genetic or phylogenetic questions with Goodeids. And so this is basically the first study that we completed on Goodeids, and at the very end I'll talk briefly about a couple of projects, ongoing projects that we have with Goodeids as well. So this is a project conducted in collaboration with my graduate student, Karis Kenway. This was part of her master's work at Southeastern Louisiana University, where I'm a faculty member. And of course, Omar is involved in, I think, most of the Goodeids studies phylogenetically and population genetically, so I want to acknowledge his contribution as well. So I'm going to focus on Xenotoka eiseni, the red-tailed splitfin, talking about phylogeny, conservation, and taxonomy. So I'm going to step way back to the beginning. Carl Linnaeus, very famous botanist, in 1758 published this manuscript, Systema Natura, where he attempted to list all of the organisms on planet Earth at the time. And I believe there was about 4,000 species that he listed at that time. And really the main contribution that came out of this work was his classification, Linnaean classification, and binomial nomenclature, such that there are no valid taxonomic names that are older than 1758. So his major contribution was what we refer to now as the field of taxonomy. Now in his manuscript there were no Goodeids. He didn't know Goodeids at the time. The closest relative of the Goodeids that he talked about was Fundulus heteroclitus fundulid, a cyprinodontid, that we have in the U.S. So he essentially is the father of taxonomy. Later, Charles Darwin, obviously major contribution to evolution and a mechanism for how evolutionary change occurs, his idea of natural selection. In his book, he said that living species are essentially related by common ancestry. And this is actually a drawing that was from the Origin of the Species. Basically, what we derived from this was the term phylogenetics. It wasn't actually until 1966, a German entomologist, Willy Hennig, that coined the term phylogenetic systematics. And so I talk about these two scientists because their contributions are really important to the things that I do. So under the umbrella term systematics is phylogenetics. Again, originally Darwin's idea, later pioneered by Hennig, which is reconstruction of evolutionary history. And then the work of Linnaeus, the taxonomy, the science of naming and classifying organisms. He gave us rules to follow in terms of how we view organisms, how we organize organisms, such that we have nomenclature and classification under the sort of umbrella term taxonomy. Now I put this forth because this is all very important. Myself as a phylogeneticist, we don't just generate trees because we can. We generate trees to use those trees to inform classification. We use those trees to ask other questions. For example, the factors that could have contributed to speciation, diversification of a group of organisms. We can use phylogenetic trees to examine the timing of diversification. So yes, we generate trees because we're interested in them, but we use those trees to address other questions. Okay. Such that, this is basically how it works. We develop a phylogeny, and the topology, or the set of relationships among the organism's interest, can then be used to guide us in developing a classification scheme. And essentially that is one of the important aspects. Phylogenetic classification should reflect evolutionary history. This is the evolutionary history. This is the classification that is derived from that. So they work synergistically, they work together. And I'm going to hopefully show you the first part of this talk that it kind of follows this line of thought. Okay, so here's the organism of interest that this talk is based on. This is Xenotoka eiseni, discovered by Ruger in 1896. Called the red-tailed splitfin is one of the common names. Various authors have noticed population declines throughout the range. John obviously talked about that just recently. Most Goodeids are in trouble. A large portion of them are in jeopardy of extinction. These are populations, I believe these are from Tamazula or Compostela, I don't remember which one was which, and these are from the inland populations in the Magdalena Basin. This is actually one of the type specimens, which is at the California Academy of Science. So when Rutter described it in 1896, it was based on one of these specimens. A lot of museums nowadays will have photographs of the actual type specimens available online. There's actually even an X-ray of this specimen. So if you want to go there and you're interested in vertebrae, number of vertebrate species it has, you can count them from sitting in your office or sitting at home looking at the X-ray image of this type specimen. So the type locality is in the Rio Grande near Tepic, Mexico. It is the type locality for Xenotoca eiseni. Previous work done by Shane Webb and Omar and others have indicated that there is a close relationship of Xenotoca eiseni and Xenotoca melanosoma. Basically, both the phylogenies show that that is the sister, or the most closely related taxon to Xenotoca eiseni. Some of the other such relationships, these are both mitochondrial studies, but different genes, show slightly different relationships, but for the most part, these two studies are very similar to one another. Again, so we're talking about this part of Mexico, west central Mexico, essentially the entire distribution of Xenotoca eiseni. And this is a modified map from Miller's Fishes of Mexico book in 2005, showing the distribution of the range of Xenotoca eiseni in the Ayuquila, the Ayuquila, the Cahuayana, and then the inland Magdalena Basin, the Compostela, and the Rio Grande de Santiago is basically the strongholds for this species. Certainly, again, a lot of this is sort of replicated from what has previously been said, but there's a lot of ongoing anthropogenic problems in this part of Mexico. John showed some sugar refinery pollution and other issues going on. Introduced tilapia has obviously been a major problem. Pulling off of water for agricultural purposes have also caused some significant problems, extinctions, extirpations, and reductions in population size of this species and other species in this region. This is some of the habitats where Xenotoca eiseni occurs. This is the San Marcos population. I believe this is one that's in the hobby. That's one of the populations. This is what it looks like. It's not the most pristine location or locality. Essentially, it's just a very muddy ditch surrounded by lots of agriculture. This is Seistanero in the Rio Grande de Santiago. Pretty good habitat, in good shape. This is Rio Compostela. We've been there. Well, I've been there twice, and the first time we went there, Xenotoca eiseni was very common, very abundant. But 3 or 4 years later, we went there, and it wasn't. It was almost extinct. We found very few individuals. That was introduced. I don't know, the placenta there. I don't know which one it was. That wasn't there 4 or 5 years earlier. So just in that short time frame, we saw pretty significant reductions of Xenotoca eiseni at the Compostela site. Okay, so essentially we have this widespread species, occurs in multiple basins. It's disjunctly distributed. In other words, all these basins are not connected to one another anymore. And we have environmental issues in this region. So the first question I wanted to address was, are there genetically distinctive populations of Xenotoca eiseni? And this is what we call a phylogeographic approach. So when we do a phylogenetic analysis, what is often done, or historically is done, you take one population or one species, one individual, and see how it's related to all the other Gudias, for example. In a phylogeographic approach, you combine phylogeny and geography. So we take a more comprehensive survey of the population. So basically, I want to take representatives from all of the extant populations, throw them in an analysis, and see how they're related or how they're different from one another. That is phylogeography. And so previous work by Omar suggested that they are pretty diverse. Some of the populations of Xenotoca eiseni are fairly diverse from one another. For example, using cytochrome B, which is a mitochondrial marker, he found significant differences between the interior Magdalena Basin, from a Magdalena Basin population, and 1 or 2 other populations. I wanted to take that and expand on that, examining more populations than Omar had originally done. So there is some suggestion there is differences, but I wanted to further investigate that. So again, we have representatives from all the populations except for the Ayuquila. John had gotten them several years earlier, but the couple times that we had been there, we've been unable to obtain specimens from the Ayuquila. So essentially, I have them from all of the other localities where they're known in this analysis. Okay, so essentially what we do, I'm going to try and simplify the molecular part, not to scare you or anything. We extract the DNA. We take a fin clip in multiple specimens from a population. We preserve that fin clip in ethanol. We take them back to the lab. We extract the DNA. We get rid of all the proteins and other excessive material. We have simply pure DNA. We use a set of primers that allows us to take a small number that isolates a particular gene and amplifies up into hundreds of thousands and millions of copies of just that one gene. That allows us to then analyze it further. We have enough material to work with. So this is called PCR, polymerase chain reaction. And so the marker that I had chosen for this part of the study is cytochrome B, same one that Omar and Duarte and Dominguez had used in their 2005 paper. We gathered sequence data, over 1,000 base pairs of cytochrome B sequence data. This is what it looks like when you get it off of the DNA sequencer. So just a bunch of different colored peaks. And then we did a phylogenetic analysis. In this case, I'm not going to go into detail, but we did a Bayesian phylogenetic analysis, which is a model-based approach. Okay, so the results. So this is the topology that I got. So this is the result of a Bayesian phylogenetic analysis. And in this analysis, the length of the branches corresponds to how genetically divergent the individuals are from one another. So the shorter the branch, the more similar they are to one another. So essentially what I got is two divergent clades. So they're all members in this light blue clade I'm calling clade 1. They're all each other's closest relative. And here is clade 2. Clade 1 consists of populations from the Rio Compostela, Seis de Janeiro, and the Rio Tamazula. And those populations are depicted here, 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 and here. These three light blue populations correspond to these populations here. You can see there's not a lot of divergence or a lot of genetic differences within that clade. But when you compare this clade to this second clade, they're over 2.5% divergent from one another, which is significant for Gudeias. For example, the two Zoneticus species are about 3% different from one another. So within one widespread species, it's almost as divergent as these two species of Zogoneticus. The second clade consists of the inland population, the Enrique Magdalena Basin, Magdalena, San Marcos, and El Maloya populations. So two divergent clades that are each monophyletic, each other's closest relatives. And if we break it apart again, Compostela and Seis de Janeiro are in this upper clade here. Again, there's really no difference between Compostela and Seis de Janeiro. There's maybe one in 1,000 base pairs of DNA sequence data. There's one base pair difference between Seis de Janeiro and Compostela. So they're basically all the same genetically based on this analysis. Same thing goes for the inland populations. They're all very closely related to one another. And these data, these values, or the divergences here, they are strongly supported. 98% support value for that node suggests that this is a very strong relationship based on this data. So, to summarize this first part, there are essentially two deeply divergent clades of Xenotopha eiseni. One that consists of the Endorheic Lago de Magdalena populations, and then all the other river basins of the Pacific coast where eiseni occur. Two clades that are very divergent. Again, over 2.5% divergent for those two clades. There are some Gudeias within a particular genus. There's less diversity than that. So eiseni is as divergent as several other described Gudeias already. if you follow a strict molecular clock to age the divergence of cichlids, which I'll say I'm not an advocate of. There are new approaches in which we can identify the age of a particular clade. I haven't done it yet. It would suggest that clades 1 and 2 diverged about 2.8 million years ago. With some of the newer approaches which incorporate fossils, it's probably a little bit older than that. I haven't done that yet. I'm working on it right now. So about 2.5 million years, those two groups diverge from one another. Okay, so that's essentially examining variation across the range of the species, taking a very crude approach. But from a conservation perspective, we also want to know what's going on within the individual populations. So the second objective is to examine inter and intra, between and within population levels of genetic diversity for these populations. So I use the same populations, but in this case, a different set of markers and more individuals. Why is this important? Well, we need to understand genetic structure if we're going to manage and conserve this resource in the best, most informed way. We know that with a variety of other studies, both terrestrial and aquatic, habitat fragmentation can lead to a lot of negative outcomes. It disrupts gene flow among populations, particularly in terrestrial environments. You have a formerly widespread population that you sort of remove, think of a large continuous forest, and you log out the middle portion of that system. Now you have two isolated populations upon which the organism cannot, maybe not move in between anymore. Maybe the habitat is inhospitable. So you can disrupt gene flow. If this fragmentation goes on for a long enough period of time, essentially on the order of millions of years, it can eventually lead to significant genetic differentiation for those populations. And finally, one approach that's often used is the identification of evolutionary significant units. And in essence, this is kind of what many of the Gudea hobbyists, including yourself, do. You recognize populations of Eiseni from the Magdalena as a distinct population, and compared to Xenotoga eiseni from the Pacific Basin. So in essence, you kind of have already recognized evolutionary significant units. There's some variation here. Could be a color pattern, could be behavior. So this approach is actually called the use of ESUs. Distinctive populations. They're not distinctive at the species level, but distinctive populations. Okay, so again, we sampled the same populations that I had done in the previous analysis. This time it was 235 individuals from 6 different localities. This is an approach called utilizing microsatellite data. It's a different type of molecular data. It's the same type of data which police use to determine who was at a particular crime scene. It's the same type of analysis, just applied to fishes in this case. So there's 8 gene fragments that we've analyzed in this case. Variety of analysis, I'm not going to go through this. Essentially, here are some of the results. So here are the 6 populations here. Here's the number of individuals that we analyzed from each population. This is a measure of heterozygosity, or a measure of how much genetic diversity there is within a particular population. So Compostela, which we analyzed almost 60 individuals, has the lowest level of heterozygosity, the lowest level of within population level genetic diversity. This Almaloya is probably somewhat artificial because we only included 8 individuals. It's probably much lower than that. But essentially these levels are moderate. They're not high levels of heterozygosity. They're not low levels of heterozygosity or level of genetic diversity. It's moderate levels of within population level genetic diversity. Also, 2 of the populations, Seistanero, Compostela and Tamazula, show evidence of a small degree of inbreeding. So that would suggest those populations are pretty small if they're inbreeding with one another. This analysis, all I want you to know is that the higher the number, the more genetically divergent populations are from one another. So if we think of the two clades that I presented in the previous part of this talk, clade 1 and clade 2, they are highly divergent from one another. And in this analysis, based on 8 different fragments of DNA, they are also genetically divergent from one another. I can identify populations based on this analysis as well. So within clades, there's not a lot of variation, but if you compare clade 1 to clade 2, those populations, there's a lot of variation with this analysis as well. And finally, this is what's called the structure analysis. It indicates that there are the 6 populations that I examined in the study are distinctive from one another. Essentially, the purer the color, the more pure that particular population is. In other words, there hasn't been recent influence of gene flow from one population to another. So 2, Seyss Neonero is, in this case, almost all green, which suggests that it is a very pure population. Compostela is not entirely pure. It suggests that there has been some influence from the Tamazula recently, but it's still relatively pure. And this analysis says that of the 6 populations that I looked at, basically 6 of them, maybe 5 of them, are distinctive from one another. And this last analysis, this is called an FCA. It's just a different way to look at the data. Same result. I get all the individuals, or most individuals from clade 1 grouping together, and most of the individuals from clade 2 grouping together. So different analyses, different markers are basically telling me the same thing. There are 2 distinct groups of isomite, the inland form and the surrounding Pacific basin populations. Again, this is basically what I just said. El Maloya and Magdalena Basin, or Magdalena populations, are not significantly different from one another. They're in close proximity, but that's the one set of populations that really aren't that different from one another. Most of the other ones are genetically identifiable, and they are divergent from the San Marcos population. Magdalena and El Maloya are geographically proximate to one another, and there appears to be some recent gene flow. Now, I'm not talking about the last 100 years or 50 years, probably in the last several thousand years, there has been gene flow among the Magdalena and Maloya populations. And we see a similar pattern for Zoogoneticus quetzaliensis in populations in the same geographic area. So there's probably some common phenomenon that is sort of can be attributed to that recent levels of gene flow. So, if we take the ESU approach, are there distinctive populations? Which ones are they? Well, clearly, clade 1 and clade 2 are very distinct from one another. Well, if you look at within those clades, you could theoretically treat Seyss Neonero, Compostela, and Tamazula as distinctive populations from one another. And if, I don't know how many of these are actually in the hobby, but if they are, they should be maintained as separate populations. You should be mixing these in the hobby if you want to maintain pure strains. And the same thing goes for here. We have San Marcos population as one, and Magdalena and El Maloya as a second population. These could be combined. There's really no difference genetically. But between there, yes, they are genetically identifiable from one another. So what can we do? And John kind of alluded to this a little bit, so I'm kind of going to fly through this. What can we do for conservation? Well, there are 3 obvious approaches: genetic rescue, habitat protection, and then captive breeding programs. You can think of genetic rescue as you have a small, endangered population of any Goodea, for this matter, with a low rate of immigration, a low rate of individuals from other populations moving in. And that's basically what we have. All these populations are isolated from one another. There's no gene flow contemporary moving into these populations. If that was going to happen, you would have constant resupply of genetic material from one population to another, which in the long term could lead to increased fitness, more genetic diversity. That's sort of the key. The more genetic diversity, the better. An alternative approach could be to take, if there was a low rate of immigration, and say you wanted to move populations from an adjacent one of these populations, Magdalena, to San Marcos, you could do that. But many of these populations are also very small. An alternative approach, which I'm not advocating, would be to take something like this to reseed this population. You could do that if you absolutely had to. However, when that typically happens, when that is done as a last resort, it typically results in reduced fitness because these fish genetically are too different from this population. So that's kind of a last resort thing that we could do, but again, I don't think we're to that point, and I'm not an advocate of that. Obviously, habitat protection is the second one. Reduce drainage, influence, reduce agricultural use in the region, control recreational use and pollution, and obviously control the introduction of non-native species. These are ideal approaches for conservation of Goodeids, but in this part of the world, a lot of these are not entirely feasible. Ideally, this would be great, but again, all of them, we're not going to be able to do that. And then finally, where you all come involved is captive breeding. And obviously, the Goodeid Ark and the work that Mike's doing here, and all of you are doing is great. It's great to have hobbyists involved in this if there ever becomes a need for reintroduction into the wild. Now we have a genetic framework upon which to guide reintroduction efforts. All of you are involved if necessary. This is sort of a great collaboration that we could have to do this if necessary. So to summarize, Eisenite consists of two evolutionary distinctive clades, or two evolutionary distinctive groups. Within each of these groups, there are about 5 distinctive populations. Of the 6 I looked at, 5 of them are genetically identifiable from one another. What this suggests is that it's possible, it's likely, Omar thinks it's the case, that Xenotoca eisenite is actually 2 different species. The Pacific Basin streams and the interior Magdalena Basin. However, I like to take a more conservative approach, and I don't think that we should be resolving or changing the taxonomy, recognizing new species until we can examine them morphologically and find characters that allow us to identify clade 1 from clade 2. So genetic data says they are different, but we really shouldn't be changing names and describing things until we have morphological characters to support those designations. And so clearly conservation needs to be done. These are 2 of the habitats. John and I have been in the same places, so we have all the same photographs. This is El Maloyo. It's a concrete lined swimming hole. And Magdalena, it's not a nice area. It's in not the best shape either. But conserving the habitat is far and away the most important thing that we can do to save Goodeids and the other species that co-occur with our species of interest. So, quickly to summarize, or the close up here, I have other ongoing Goodeid studies. Right now, I've been looking at phylogeography of Iliodon, kind of taking the same approach with Xenotoca, with Iliodon, examining populations from all over the range, and this is also in collaboration with Omar. Do the same thing for Goodea, multiple populations from across the range, seeing how genetically divergent they are from one another. We have some systematics, higher level systematics, and then some taxonomy of Shopalicides that I've been working on for a couple of years now. So hopefully in a year or 2 I can come back and talk about results of some of this work. But again, most of this is kind of ongoing at this point. Finally, I want to thank John in particular for piquing my interest in Goodeid work and taking me to Mexico all these years. And finally, Karis, Kenway, and Omar as project collaborators on this study. And if Google Translate is correct, then I hope I covered most of the languages that are here.
In French.
And I'll take any questions.
You didn't yet incorporate morphometrics to try to support these 2 clusters that you.
That's the next step, is to look at morphology to take it to the next level.
But you I guess you saw most of the specimens that you really use for the genetic studies.
Yeah.
Do you have a general impression if the phenotypes maybe support this?
I don't, because I haven't looked at the specimens in detail. A lot of this, John and I collected a lot of these fish, but Omar provided a lot of the samples as well. So he has a lot of the voucher specimens, the morphological specimens in Michoacan, which I haven't seen yet. So the answer is, I don't know how they differ morphologically. That's something Omar and I are talking about now.
I understand that there are here some old strains in the hobby for which you don't have an exact locality. Could you use this approach to work backwards and connect them with at least?
If I had access to one specimen, I could tell you it's clade 1 or clade 2. If I had access to multiple individuals, 10, 15, 20 individuals, then I could be more refined answer of which population. Because the second one requires a lot of individuals to do those analyses.
Yeah, but these old strains are probably highly inbred. So I expected they lost a lot of genetic diversity.
Yeah, I mean, if they're really, if you
count
think,
them as well.
yeah, if you think they're really inbred, then probably the best that I could do is clade 1 or clade 2. That's probably about the best that I could do if you think they're really inbred.
Yeah, but that's maybe could also say morphologically in the end.
Yeah, maybe morphologically as well once we figure out what the differences are. Absolutely.
You have the next generation
Uh-huh. Yeah. So he asked, there's a new, which I haven't talked about, there's a new genetic approach called next generation sequencing. So the stuff that we do now is called Sanger sequencing. We analyze one gene fragment at a time. This next generation sequencing approach allows you to gather data at one time for 300, 400, 500 plus gene fragments at one time. So basically it's more of a entire genome approach at gathering genetic data. So he asked if I had that data, would I still need to do the morphology to determine whether they're different or not? I'm more conservative because I go out in the field and I sample these fish. I want to know how I tell this one from this one. So my perspective is I feel you need to have at least one diagnostic morphological character that separates this population from that one. Certainly the next gen stuff is going to give us a whole new perspective on a lot of things, relationships and divergences. But I personally think you have to have morphology to diagnose something as a different species. Okay?